let's see how a two-trait cross is done. Let's work through a Punnett square for a two-trait cross. Now, hopefully you've already done the practice problems that go along with the one-trait cross. You're feeling very comfortable doing those sort of smaller Punnett squares. I'd like to show an example of how we do, how we show two traits being inherited together, do that 16 square Punnett square, and get to the important piece about interpreting the data. So what we're going to be doing for our two-trait cross examples, we're going to be following with the pea plants again. And just a reminder, it's not that I really love peas or anything. It's that when we're looking at the pea plants, we're following some of Gregor Mendel's work. And when it comes to the field of genetics, we didn't have terms like dominant and recessive until we had Gregor Mendel's work. And the reason why he was working with pea plants is because there were so many. He was a statistician, so he could get these beautiful data sets. And with pea plants, we have this ability for one gene to make one trait, so it was really, really straightforward. Towards the end of these lecture slides and getting into part 2 of genetics, we get into some of the more complicated patterns of inheritance, get more closer to reality. So we want to stay really focused with these straightforward examples. We've already seen how the trait of height is being inherited with pea plants. Remember, our phenotype of being tall is dominant to that phenotype of being short, that is recessive. And we already have the example in the lecture slides of pea pod color. When it comes to pea pod color, the green pea pod is dominant and the recessive is yellow. Now, one of the interesting things you may or may not have ever consciously recognized is when you go to the grocery store, like almost all of your pea pods are going to have this green color. You never, ever see anything else. But we know that within peas, it's possible to have this yellow pod color. And researchers are always experimenting and playing around with things. And the question is, why would we care about these traits? Well, one of the things that I used to do is I used to work as a farmer. I used to grow things for a farmer's market. And if you have something that's new, something that's unusual and unique, you can charge more money. So a new development are these purple pea pods. Nobody has them. So not only do we have the traditional green and the yellow that we're going to talk about today, but there's also a possibility for purple. Now, if I'm working in a farmer's market, I know this year I could charge a ton of money for this because nobody's got it. It's unusual. It's going to make culinary dishes pretty. If I was working and partnering with restaurants, I would go to them specifically and say, I've got this new product coming in. This is going to be great for your restaurant. So let's say I'm a farmer, and what I really want is I want that unique, unusual coloration that nobody else has got. In this case, yellow pea pods. But having been a farmer, I can tell you, you're going to want the tall plants, because after harvesting hundreds and hundreds of pea pods, bending over for short plants is just going to kill your back. So you're going to want to grow tall plants that are making that unusual pea pod. So remember, with our Punnett square, we have our 4 main steps. Our first step is we have to figure out our parental or pea generation. So in our pea generation, let's say we have a female, and that female is heterozygous for height. She also has a heterozygous genotype for pod color. We have our 2 separate letters. The letter T is being used to represent height, and the letter G is being used to represent pod color. These 4 letters represent our 2 separate homologous pairs of chromosomes. We want to see how these traits are being inherited together. Let's say we have a male who also has a heterozygous genotype. Heterozygous for height and heterozygous for pod color. Our next step is we have to model meiosis. When we're going to model meiosis, this is when we're going to set up that 16 square Punnett square. Now, it doesn't matter if our female's gametes go on the top or if our female's gametes go on the side. Either way, we're still going to end up with the same combinations in the middle of our square. In other words, the same combinations for our diploid zygote or offspring. So I'm going to stick with the idea of keeping the female's gametes up here on the top. What I next need to figure out is all of the possible combinations of height, letter T, and pod color, letter G, in each of these gametes. We're modeling meiosis. I'm starting with a diploid cell with 4 letters. In each of my possible gametes, my gametes are haploid. I have to have half of the letters, so I should only have 2 letters, 1 letter T and 1 letter G. In order to figure this out, I'm going to use the FOIL method. I'll take the first letter from each homologous pair of chromosomes. So one possibility for my female's gametes is that she has the dominant gene for height and the dominant gene for pod color. I take the outside letter from each homologous pair of chromosomes. Another possibility is the dominant gene for height, but that recessive lowercase letter for pod color. I take the inside piece of DNA from each homologous pair. Another possible combination is recessive for height, but dominant for pod color. And then I take that last letter from each homologous pair. So my final possible combination is recessive for height and recessive for pod color. The next step that I have to do is I have to figure out all of the possible combinations of genes or alleles for my male's gametes. I'm going to FOIL my male's genotype in order to figure out all of those combinations. Take the first letter from each homologous pair. Take the outside letter from each homologous pair. Inside letter. And last letter. So now that I have all of the possible combinations of genes in my gametes, the next step is to make the diploid zygote, to make the offspring. I like to keep my letters together. So I'm keeping my T's together for height and my G's together for pod color. The reason why I like to do this is because visually it'll make it easier for me to pick out those phenotype combinations. Being tall and making green pea pods, being tall and making yellow, being short and making green, or being short and making yellow. When I have a heterozygous genotype, I'm going to stick with that pattern, with that etiquette of putting the capital letter first. This way, again, makes it easier on my eyes to pick out the phenotype combinations. Now our next step is to look into our data, look into this Punnett square, and to figure out the probabilities. Now I could go through and list all of the possible genotypes and their possibilities, but typically when we do this, because every possible combination can happen for genotypes, we skip ahead with this type of Punnett square and go right to interpreting the phenotypes and their probabilities. So one phenotype that I'm looking for, phenotype combination, is I'm looking for how frequently will my offspring, my diploid zygote, show me the dominant trait for height which I'm going to abbreviate with H, and the dominant phenotype for pod color. So how frequently will I find both of those traits together? If I look at my Punnett square, I'm looking for capital letter T's and capital letter G's to show me dominant height and dominant pod color. So I have 1, 2, 3, 4, 5, 6, 7, 8, 9. There's my 9. My next phenotype combination is how frequently will I see dominant height, but that wonderful recessive yellow pod color? How frequently will I have those tall plants giving me that unusual pea pod that I can charge more money for in the farmer's market? So I'm looking for capital letter T's for height, but I need a homozygous recessive genotype. I need both lowercase letter G's in order to see those yellow pea pods. So I have 1, 2, 3 out of my 16 squares where that's a possibility. My next combination is how frequently will I see the recessive height being short and also having that dominant green pea pod. So I'm looking for lowercase letter G's, lowercase letter T's, but capital letter G's, excuse me. So I'm looking for that homozygous recessive genotype for height, but I need at least one capital letter to show me that dominant green pea pod. I have 3 out of 16 squares that will show me that. And then finally, how frequently will I have plants that are recessive for height, so they're short and are showing us that yellow recessive pod color. Excuse me, kitty. In other words, how frequently will I see that double recessive, homozygous recessive for height, and homozygous recessive genotype for pod color? Only 1 out of 16 squares will show double recessive. So hopefully that helps you to understand the 2-trait cross, how we do it, and how we interpret the data. Don't forget to finish those practice problems in your packet. And I know some of you have asked about some of the animals that make the cameo appearances. This is Melgus, and he has been enjoying the eraser and enjoying helping us today. Email me as always if you have any questions.